Hello everyone, I am Payal Pridrashni, PGT Biotech, Kendriya Vidyalaya number 3, Delhi Cant. So today I am going to make you learn about transcription. Before going more in detail for transcription, let us answer the question which I asked in my earlier session. So my first question was, why DNA replication is required? For all living system, for all living cell, for all living organism, DNA replication is very much important process. So why it is required? So answer is, DNA replication is required for cell division and reproduction. If DNA replication would not be there, then after each division the number of chromosome will get reduced and ultimately the cell will die. So to maintain the consistency of chromosome after each division, the DNA replication is required. The next question which I asked, which enzyme is responsible for polymerization of DNA? Polymerization of DNA means how the nucleotides are being added up, or we can say how the DNA is being replicated, and the answer is DNA polymerase enzyme. DNA polymerase enzyme is an enzyme which helps in DNA replication, or we can say polymerization of DNA. If we talk about bacterial DNA replication, more particularly DNA polymerase 1 and DNA polymerase 3 helps in polymerization of DNA, and in case of eukaryotic cells, DNA polymerase alpha and DNA polymerase delta, both these DNA polymerase help in polymerization of DNA. The next question is, what is the direction of DNA replication and why? In my earlier session, I told you the direction of replication is 5 to 3 prime. So the answer of this question is, the direction of replication is 5 prime to 3 prime. Now the another question, why 5 prime to 3 prime? Why not 3 prime to 5 prime? So the answer lies in the structure of nucleotide. A nucleotide is having one nitrogenous base at first number of carbon, one sugar molecule and phosphate molecule at fifth number of carbon. At the second number of carbon, the DNA is having hydrogen, and at third number of carbon, the nucleotide is having OH group, and because of the presence of free OH group, the DNA polymerase is able to add the coming nucleotides at 3 end only. So the direction of polymerization or replication would be 5 to 3. The next question which I asked was, what are Okazaki fragments? As we learned in the process of DNA replication, both the strands are being copied. One of the strands are copied in a continuous manner, and that strand is called leading strand, and the another strand is being copied in a discontinuous manner, and that strand is called lagging strand. So in lagging strand, the DNA are synthesized in fragments, and these newly synthesized DNA fragments on lagging strand is called Okazaki fragment. Now let us answer some of the question of your textbook exercise. The first question, group the following as nitrogenous base and nucleoside: adenine, cytidine, thymine, guanosine, uracil and cytosine. You have been given these names and you have to identify which are nitrogenous bases and which are nucleoside. So, you have learned that there are 5 types of nitrogenous bases are there. The first one is adenine, second one is thymine, third one is cytosine, fourth one is guanine and fifth one is uracil. These 4 adenine, thymine and cytosine are found in DNA, and leaving the thymine, remaining adenine, cytosine, guanine and uracil are found in RNA. So, RNA contains adenine, cytosine, guanine and uracil. So, these are nitrogenous bases, and when these nitrogenous bases combines with sugar, they form nucleoside. So, again there are 5 types of nucleoside found in nucleic acid: adenosine, thymidine, cytidine, guanosine and uridine. So, we can easily identify which are nitrogenous bases and which are nucleoside. So, nitrogenous bases are adenine, thymine, uracil and cytosine, and nucleosides are cytidine and guanosine. The next question is, if a double stranded DNA has 20% of cytosine, calculate the percent of adenine in the DNA. So, here you can use the Chargaff rule. According to Chargaff rule states that the number of purine in particular DNA is always equal to number of pyrimidine. So, here if 20% is cytosine, then 20% guanine would be there. So, overall they will form 40% of the DNA. So, out of 100, if 40% is being removed, so remaining 60% would be adenine plus thymine. So, the number of adenine and thymine are equal. So, if we divide 60 into 2 by 2, it will give 30. So, 30% adenine would be there if a DNA strand is having 20% of cytosine. The next question, if the sequence of one strand of DNA is written as follows, write down the sequence of complementary strand in 5' to 3' direction. For this, you have to recall the complementary sequences. For adenine nucleotide, the complementary is thymine nucleotide, and for cytosine, the complementary is guanine nucleotide, and vice versa. So, with the help of this complementary, we can find out the complementary sequence of this particular DNA, but while writing the complementary sequence, we will start from any of the ends, but the answer you will get is that will be in 3' to 5'. So, you have to write the answer in 5' to 3'. So, answer will be this one. The next question is, depending upon the chemical nature of the template DNA or RNA and the nature of nucleic acid synthesized from it DNA or RNA, list the types of nucleic acid polymerases. So, if we talk about the polymerase enzyme for DNA or RNA, the answer is there are 2 different types of nucleic acid polymerases. One is DNA dependent DNA polymerase enzyme which help in polymerization of DNA, and second one is DNA template RNA polymerases which helps in RNA polymerization or transcription. Now, let us start the today's session, that is transcription. So, what is transcription? Do you know? Yes, transcription is transfer of genetic information from DNA to RNA, or we can say synthesis of an RNA copy from DNA. Now, what does it mean? That means that there are certain genetic information which are there in the DNA which are being transferred to RNA. Now, what is this genetic information which is there in the DNA? This genetic information is nothing but it is protein, and with the help of this central dogma we will be able to understand why transcription is necessary. You can see over here the DNA is being copied to form DNA, and that is called replication. This DNA will be processed by the process of transcription to form the RNA, and this RNA, particularly messenger RNA, will undergo the process of translation to protein, synthesize the protein, and the main worker of our cell is the proteins. So, to synthesize the protein, transcription is very much required, and RNA only will give the codes for the amino acid to synthesize the protein, which we will discuss in my coming session of protein synthesis. Now, the important question, direction of transcription. As we learn in replication, the direction of DNA replication is always 5 prime to 3 prime. Here also the direction of transcription is 5 prime to 3 prime, and the answer is why 5 prime to 3 prime lies in the structure of nucleotides which forms the RNA. So, in case of RNA, the sugar is ribose sugar. At the 5th number of carbon, they have phosphate group. At first number of carbon, they have nitrogenous base, and at second number of carbon and third number of carbon, they have OH group. In case of DNA, at second number of carbon, OH is not there, hydrogen remains there, that is why it is known as deoxyribose sugar. But in case of RNA, ribose sugar is there, and in ribose sugar at second number and third number of carbon they have OH group, and RNA polymerase is able to add the nucleotides at third number of carbon having the free OH group to form the phosphodiester bond, and because of this we can find the direction of transcription will be 5 prime to 3 prime. So, I think the question will be very much clear for you. Now, what are the different types of RNA which are being synthesized by transcription? Here we will mainly focus on 3 types of RNA, but there are so many RNAs are there which involves in different processes, and these are messenger RNA. Messenger RNA gives the codes or gives the signal for protein synthesis during the process of translation. Second one is ribosomal RNA, which help in formation of ribosomes, and the ribosomes help in protein synthesis during translation by reading the messenger RNA sequences. The third one is transfer RNA, which brings the amino acid to the messenger RNA whenever the signals are given by the transfer RNA during protein synthesis. So, all these 3 RNAs help directly or indirectly in protein synthesis. Apart from these 3, some more RNAs are there which involve in modification and processing of RNA. These are a small nuclear RNA known as snRNA, a small nucleolar RNA known as snORNA, and a small cytoplasmic RNA as scRNA. So, these are few types of RNA which are formed after transcription. But you must know about this, that like that of replication, the replication occurs in whole the strands of the DNA. That means whatever DNA is there, the whole DNA will be replicated, but in case of transcription only few fragments of DNA will be transcribed. The whole DNA is not being transcribed to form the RNA. So, transcription is very selective process, a particular fragment which will synthesize the messenger RNA or ribosomal RNA or transfer RNA or any other RNA will be transcribed only, rest DNA will remain as such. Now, what are the requirements for transcription? The first is DNA template. What is this DNA template? So, if we talk about DNA template, you might be knowing DNA is double stranded helical structure, and this double stranded DNA will be copied to form single stranded RNA. Now, here lies an important question: how does double stranded DNA will be copied to form single stranded RNA? Why not double stranded RNA if DNA is double stranded? So, the answer is that only one strand of the DNA will be transcribed during transcription, and as I told you earlier, RNA will be synthesized in 5 to 3 prime. So, only one strand of this particular DNA will be transcribed to synthesize single stranded RNA. So, we need a single stranded RNA during transcription so that the protein synthesis can be done. For the transcription, we need single stranded DNA which is known as template, which will be copied. So, template is an important requirement for transcription. Next is the raw materials or substrate needed to build a new RNA molecule. We need substrate which will polymerize together to form the RNA molecule. And the third, that is transcription apparatus. These include certain proteins, certain enzymes to synthesize the RNA molecule. So, which strand of DNA will be transcribed? This is an important question. We always do the mistake while writing the ends of the DNA or RNA or the base sequences. So, if we talk about the DNA strand which will be transcribed, so this will always be 3 prime to 5 prime, because the DNA is having 2 strands, one will be 5 prime to 3 prime and another will be 3 prime to 5 prime. So, transcription will be in 5 prime to 3 prime end, and the RNA and the template which is going to transcribe both will be anti-parallel. So, if RNA is 5 prime to 3 prime, so DNA which will be transcribed will be 3 prime to 5 prime. So, always 3 prime to 5 prime DNA strand will be transcribed to form RNA molecule. Now, substrate for transcription. As I told you earlier, there are 4 types of ribonucleotide triphosphate, which includes riboguanosine triphosphate, riboadenosine triphosphate, ribocytidine triphosphate and ribouridine triphosphate. These nucleotides are required for the polymerization of RNA, or we can say for the process of transcription. And the third requirement, the transcription apparatus or the enzyme which involve in transcription is RNA polymerase. RNA polymerase basically have 4 subunits, or we can say 4 polypeptide chain. 2 polypeptide chain are of alpha, 1 is of beta and 4th one is of beta prime. So, these 4 subunits combines together to form core enzyme which is known as RNA polymerase. With this core enzyme, a sigma factor is being attached which gives holo enzyme, and this holoenzyme is responsible, or we can say it helps in the initiation of transcription process. Now, what are the functions of RNA polymerase enzyme? The first, it searches and binds to the promoter. During transcription, it is very much difficult for RNA polymerase to bind over DNA molecule, because RNA polymerase would not be able to recognize the transcription start site. So, the RNA polymerase with the help of sigma factor recognizes the promoter and binds over there. The next function of RNA polymerase is it unwinds the short stretch of double helical DNA. As we know, we need a template, DNA template, which is going to be transcribed, and for getting the template, both the strands of the DNA needs to be unbinds, and for unbinding this RNA polymerase helps. The third function of RNA polymerase is catalyze the formation of RNA. After unbinding the double stranded DNA, it will help in the formation of RNA. The RNA polymerase detects termination signal. If a certain process has been started, it has to be terminated also. So, for termination, the RNA polymerase detects the termination sequences and gives the signal for termination of transcription. Last but not the least, the RNA polymerase interact with repressor and activator to regulate transcription. The process of transcription is always being regulated by different enzymes or proteins. So, RNA polymerase with the help of repressor and activator controls or regulate the transcription. So, these are few functions of RNA polymerase. Here you can see the organization of DNA. Here we have RNA coding region, which will actually going to transcribe to synthesize a particular type of RNA. It may be messenger RNA, ribosomal RNA or transfer RNA. The transcription is going to start from a particular site, which is known as transcription start site, and all the nitrogenous sequences, all the base pairs which are there in the left side of this transcription start site is called as upstream, and all the sequences from this transcription start site which lies in the right side is known as downstream. To this upstream of transcription start site, there is a promoter which is recognized by RNA polymerase. So, you can know much more about this particular organization of DNA in case of bacteria, considering this segment of DNA as RNA coding region, means this particular region of DNA is going to transcribe to synthesize any type of RNA. This particular part or place in DNA is called transcription start site or initiation site, from where the transcription will begin. To the left of this transcription start site, all sequences are known to be upstream, and to the upstream to this transcription start site, these particular fragments of DNA is called promoter. This promoter is very much important part of transcription, because the RNA polymerase is able to recognize the promoter sequences, and then only it will bind over there to initiate the transcription. Now, the sequences, base sequences in the right side of the transcription start site is called downstream, and in downstream RNA coding region is there, and to the downstream of RNA coding region, this particular fragment is termination site. When RNA polymerase will reach over the transcription termination site, it will give the signal for terminating the transcription and the transcription will be terminated. So, this is how the DNA which is going to transcribe to synthesize particular type of RNA is being organized. Now, process of bacterial transcription. We are taking the bacteria as a model organism to study any biological process. So, if we talk about prokaryotic and eukaryotic transcription, both the cells have very common process of transcription. The differences are also there which we learn, which we will know in the later session. So, the first process of bacterial transcription is initiation, means the transcription process will be initiated. Second process is elongation. After the initiation of transcription, the RNA chain will be elongated, and the third one is termination. Once the RNA has been synthesized properly, the process of transcription will be terminated. So, these are three different steps or process of bacterial transcription. Now, let us talk about initiation of transcription. So, initiation of transcription begins with the promoter recognition. That I already told you कि in the upstream of transcription start site, promoter is there, and promoters have certain sequences of nucleotide which are known as consensus sequences. These consensus sequences is being recognized by sigma factor of RNA polymerase. After the recognition by the sigma factor, the RNA polymerase is able to bind over the promoter. In case of bacterial DNA, there are two consensus sequences present at -10 and -35. -10 means the sequences to the upstream of transcription start site is given the negative numbers, and the sequences to the right, that is downstream sequences, are given the positive numbers. So, at -10 there is consensus sequence, that is TATAAT, consensus sequence is there, and at -35 TTGACA consensus sequence is there. Consensus sequences means the sequences of base which are repeatedly occurring at the same position in most of the living organisms in case of prokaryotic cell. So, in case of bacteria, these two are considered as consensus sequences, and these consensus sequences are very much important for the recognition of promoter by the sigma factor to initiate the transcription. So, initiation of transcription also includes formation of the transcription bubble. So, you can see over here, for bacterial transcription, if this is a DNA which is going to transcribed, and here is the transcription start site, and this is the direction of transcription, it means this particular fragment has to get transcribed. So, to this upstream promoter is there, that promoter will be recognized by the sigma factor. This particular structure is known as sigma factor, which is present in RNA polymerase, and the whole this structure is holoenzyme along with core enzyme, that is RNA polymerase and sigma factor. So, with the help of this sigma factor, RNA polymerase is able to recognize the promoter because of the presence of consensus sequence in the promoter. When the RNA polymerase bind to the DNA at the promoter, the DNA structure is remain as such, there is no any change in the DNA structure, and this particular stage of transcription is called closed promoter complex, because this promoter sequences, the DNA of this promoter sequences is closed, it is not opened. Now, when this RNA polymerase binds tightly with the DNA, because of that both the strands of the DNA get separated out to form the template, and this particular stage of transcription is called open promoter complex. From here the transcription will be started. Now again, which strand of DNA will be transcribed? If RNA is going to synthesize in 5 to 3 prime direction, then the 3 prime to 5 prime strand of the DNA will be transcribed. So this strand will be transcribed, and the RNA polymerase without the help of primer can add the nucleotides of RNA and will initiate the transcription. So if we talk about DNA polymerase and RNA polymerase, there is a big difference between these two enzymes. DNA polymerase always need a primer that is short segments of oligonucleotides which is single stranded and which gives 3 prime end to the DNA polymerase to add the nucleotides. But in case of RNA polymerase, the RNA polymerase do not need any primers, it can add the nucleotides by reading the complementary sequences of the template. The next step of initiation is creation of the first bond between ribonucleoside triphosphate. After the separation of both the strands of the DNA, the RNA polymerase will read the sequences of the DNA and will add the nucleotides for RNA synthesis. And then escape of the transcription apparatus from the promoter. Once the addition of nucleotides has been started, these RNA polymerase will escape the promoter, it will move towards the RNA coding sequences and it will add the nucleotides to synthesize the RNA. But after the formation of 7 to 9 nucleotides of RNA molecule, this sigma factor is released and the remaining transcription is being carried out by the core enzyme of RNA polymerase. So this is the organization of promoter in bacterial DNA. This I already told you. Elongation of RNA chain, it includes RNA polymerase begins to move along the DNA. This RNA polymerase will move towards the RNA coding sequences, will read the DNA sequences and will add the RNA sequences. Sigma factor is released during elongation, this sigma factor is being released and the core enzyme alone is able to complete transcription of gene, and this core enzyme without sigma factor will move ahead towards the RNA coding sequences and it will synthesize the RNA in 5 prime to 3 prime direction. Proofreading, an important part of transcription. Proofreading means whenever there is insertion of any wrong nucleotide during transcription, the RNA polymerase itself performs the proofreading mechanism. It removes the incorrect base added while transcription using its exonuclease activity. RNA polymerase at the same time perform its exonuclease activity, it will remove the wrong one and it will add the correct one. This is called proofreading mechanism. This proofreading mechanism is also there in DNA replication. The last step of the transcription process is termination of transcription. How the transcription is being terminated? Is there any particular way or it is terminated as such? So there are two ways of termination of transcription. The one is row independent terminator, which are able to cause the end of transcription in the absence of row. Row is a protein which helps in transcription termination. If row involves in termination of transcription, then that is called as row dependent terminator. In this terminator, the row protein involves and the transcription is being terminated. If the transcription is being terminated without the help of row protein, that is called as row independent terminator sequences, and if the termination is carrying out with the help of row protein, it is known as row dependent terminator sequences. So, in case of bacteria these two type of termination is there, which is not there in the eukaryotic transcription process, which we will discuss later. So, how these two type of terminator sequences help in the termination of transcription. So, if we talk about row independent terminator, they have certain inverted repeats. Now, what is this inverted repeats? Inverted repeats means if a particular nucleotide sequences, say suppose there is A, C, T, T, A, C. So, complementary of this will be T, G, A, T and G. In certain fragments of DNA, if this particular base are there, and similarly this sequences is also there at certain another point of the DNA, A, C, T, T, A, C. Complementary of this G, T, A, G, T. So, if you see these two fragments A, C, T, T, A, C, A, C, T, T, A, C, both are inverted repeats. I hope inverted repeats will be clear to you. So, sequences are same. So, in case of row independent terminator sequences, the terminator sequence have inverted repeats. These inverted repeats have 16 to 20 base pair upstream of the transcription termination point. Upstream means left side to the transcription termination point. These inverted repeats is followed by 4 to 8 80 base pairs, and when these inverted repeats form the RNA transcript after transcription, because of these complementary nature they will form a hairpin structure. You can see over here the row independent termination in bacteria. These fragments are inverted repeats having the same sequences. So, when this particular DNA fragment is going to transcribe, it will form the RNA, and after transcription they have this type of sequences. So, if you look these sequences are complementary to each other, and because of the complementary nature they will form a hairpin structure, and because of this hairpin structure the RNA polymerase will slow down and will pause the RNA synthesis. So, this hairpin structure results because of the complementary nature of the RNA, which is synthesized by row independent terminator sequences, and because of this termination hairpin structure the transcription is being terminated. The next type of termination is row dependent terminator. In case of row dependent terminator, if we talk about the particular sequences of the row dependent terminator, the row dependent terminators are rich in cytosine nucleotide and poor in guanidine nucleotides. There are no any inverted repeats in the row dependent terminators, and because of that no hairpin structure are being formed. Row protein bind to the C rich terminator, as the row dependent terminators are rich in cytosine. So, the row will bind over the C rich terminator, and row is having a property of helicase enzyme, which we learn in DNA replication. Helicase unbinds the strands, both the strands of the DNA. So, here also the row protein acts like a helicase enzyme, it unbinds the DNA RNA helical structure, and because of that unbinding the transcription is being terminated. But the row protein take the help of ATP to get the energy, and because with the help of that energy the DNA RNA helix are being separated and termination of transcription is completed. So, this is how the process of transcription is being completed in the case of bacterial cell. So, let us recall all the concept which we learned till. Transcription is selective process. Transcription is selective process means what? Only few fragments, only certain fragments of the DNA will be transcribed to form the particular type of RNA. That means, as that of DNA replication, whole the DNA is being replicated, but in case of transcription, whole the DNA is not participating in transcription. Only few fragments of DNA will be transcribed to form messenger RNA or ribosomal RNA or transfer RNA or any other RNA. RNA is transcribed from single stranded DNA. The process of transcription needs a template, and that template is provided by single stranded DNA. So, for transcription a DNA template is required. RNTPs are used as substrate. These RNTPs are used as substrate to polymerize the RNA sequences. RNA molecules are antiparallel and complementary to the DNA template. So, as I told you, if RNA is synthesized in 5 prime to 3 prime direction, so the DNA strand 3 prime to 5 prime will be transcribed. So, both DNA strand and RNA will be antiparallel in nature and both will be complementary, but the major difference is in case of DNA they have thymine nucleotide, but while adding the nucleotides complementary to thymine there is adenine, and if there is adenine nucleotide in DNA, the complementary of adenine will be uracil nucleotide. So, this is the difference between DNA and RNA. Transcription depends on RNA polymerase. Without RNA polymerase, transcription cannot be completed. This is the enzyme which helps in formation of RNA and perform many functions during the transcription. Core enzymes require sigma factor. RNA polymerase is considered as core enzyme and it requires sigma factor. Sigma factor is very much important part of RNA polymerase enzyme because it helps RNA polymerase enzyme to recognize the sigma factor of DNA, and this sigma factor will recognize the promoter of the DNA to bind over the promoter to initiate the transcription. Promoters contain consensus sequences. Sigma factor is able to recognize promoter only because of the presence of consensus sequences, and these consensus sequences are present in most of the prokaryotic cells at the same position. RNA polymerase binds to promoter. After the recognition of promoter, the RNA polymerase binds over the promoter. Sigma factor is released during elongation when the 7 to 9 nucleotides of RNA is being added up. After that the sigma factor is released and the remaining process is completed by the core enzyme of RNA polymerase, and then the transcription is terminated with the help of Rho protein which is known as Rho dependent, or without the help of Rho protein which is known as Rho independent termination. So, this is all about transcription process in bacterial cell. If we talk about transcription in eukaryotes, there are so many similarities in bacterial as well as in eukaryotic transcription process. Like bacterial cell need RNA polymerase for transcription, there is requirement of promoter, there is requirement of template, there is requirement of substrate. Similarly, the eukaryotic cells need a DNA template, need substrate, need some certain enzymes for transcription, but there are some few differences which we are going to learn over here. So, if we talk about the differences, the major difference lie in the structure of bacteria, which is a prokaryotic cell, and a eukaryotic cell, is the presence of nuclear membrane. Consider this as prokaryotic cell, or we can say bacterial cell. The bacterial cell is not having any nuclear membrane to enclose the genetic material. So, they have genetic material in their cytoplasm. But in case of eukaryotic cell, eukaryote means they have a true nucleus, and this nucleus encloses the genetic material, that is DNA, and DNA is present inside this nucleus. So, in case of prokaryotic cell, replication, transcription, and translation all these process occurs in the cytoplasm only. But in case of eukaryotic cell, because of the presence of nuclear membrane, because of the presence of nucleus, the replication process, the transcription, is carried out inside the nucleus, and then these RNA is being transferred to the cytoplasm, and then these RNA, if it is messenger RNA, will undergo translation to synthesize protein in cytoplasm. So, this is a major difference between prokaryotic and eukaryotic cell. In case of prokaryotic cell, replication, transcription and translation, all these process occurs inside the cytoplasm. This is the cytoplasm of the prokaryotic cell. But in case of eukaryote, the presence of nuclear membrane, the presence of nucleus separate out the protein synthesis or translation from the transcription and replication. The replication and transcription occurs inside the nucleus, and then the RNA is transported to the cytoplasm, and here the protein synthesis is being completed. So, this is the nucleus of the eukaryotic cell, and this one is cytoplasm. So, here I have written, after the process of transcription, pre-RNA. Pre-RNA means primary RNA. What is the meaning of the pre-RNA or primary RNA? In case of eukaryotic DNA, they have some functional sequences which is known as exon, and they have some non-functional sequences which is known as introns. So, when this DNA is undergo transcription, they synthesize the RNA transcript having the exon functional part and intron non-functional part. So, it is not in total a mature RNA or biological active RNA, that is why it is considered as pre-RNA or primary RNA. This pre-RNA is having the sequences of exon and introns. Now, by the process of processing and splicing, the introns are being removed because introns are non-functional sequences, and all the exons will splice together, bind together to form the mature RNA. This mature RNA is functional RNA, will be transported to cytoplasm, and in the cytoplasm the mature RNA, if it is messenger RNA, will undergo translation to synthesize the protein. So, this is a major difference between prokaryotic transcription and eukaryotic transcription. In case of eukaryotic transcription, after the process of transcription, pre-RNA is synthesized which have functional as well as non-functional sequences of RNA, and then the non-functional sequences, that is introns, are being removed, and the functional parts, that is exons, are being spliced to form the mature functional RNA, and then they are being translated to form active protein. Now, here lies an important question which we learned during the packaging of DNA. In case of eukaryotic DNA or eukaryotic chromosome or DNA pack themselves in the nucleus with the help of nucleosome or histone proteins. Now, what about these histone proteins? As we know, on the histone octamer the DNA is being wrapped. So, if DNA is being wrapped over the histone octamer to form the nucleosome, how the transcription will be performed inside the cell? So, now these histones are removed by two different enzymes. One is acetyltransferase enzyme, which will add the acetyl group to the histone and it will destabilize the histone, and the histone will be removed and the DNA will be separated out. It will be free from the histone octamer. And second one is chromatin remodeling protein, which also help to remove the histone from the nucleosome model, or when the histone is being removed, the DNA is free to participate in transcription. So, this is an important aspect regarding the transcription of eukaryotic DNA. Now, let us talk about the enzyme which is involved in eukaryotic DNA transcription. As we learn in bacterial cell, there is just one type of RNA polymerase which can transcribe all the three types of RNA transcript, that is messenger RNA, ribosomal RNA and transfer RNA. In case of prokaryotic cell, only one RNA polymerase is there which is able to transcribe all the three types of RNA. But in case of eukaryotic DNA transcription, there are three different types of RNA polymerases which involve in transcription. The first one is RNA polymerase 1, which is located in nucleolus and catalyze the synthesis of ribosomal RNA. So, RNA polymerase 1 is enabled to synthesize ribosomal RNA only. It cannot transcribe, it cannot synthesize transfer RNA or messenger RNA. Second RNA polymerase is RNA polymerase 2, which is located in nucleoplasm and catalyze the synthesis of messenger RNA and some of the small nucleolar RNA. So, messenger RNA, which codes for the amino acid, which codes for protein synthesis, are being synthesized with the help of RNA polymerase 2 in case of eukaryotic cell. And the last RNA polymerase is RNA polymerase 3, which is located in nucleoplasm and catalyze the synthesis of transfer RNA and a small nuclear RNA. So, transfer RNA is synthesized with the help of RNA polymerase 3. So, if we talk about the difference between bacterial transcription and eukaryotic transcription, in case of bacterial transcription, there is only one RNA polymerase which can transcribe all the three types of RNA, and in case of eukaryotic cell, there are three different RNA polymerase which will transcribe the RNA in a different way. RNA polymerase will transcribe to form ribosomal RNA, RNA polymerase 2 will synthesize messenger RNA, and RNA polymerase 3 will synthesize transfer RNA. So, here I am going to end my session. Before leaving, let us think over some question. and try to answer this question. The answer of this question will be given in my next session. And the first question is, what do you mean by transcription? You have learned the transcription process in detail, so you might be able to answer this question. Write the RNA transcript for following DNA template. You have to write which type or which sequences of RNA will be synthesized with the help of this DNA template, and what are the differences between prokaryotic and eukaryotic transcription. Few we have discussed the differences, you can think over there. And what is the role of sigma factor in transcription? In case of bacterial transcription, the RNA polymerase is being added up by the sigma factor to form the holo enzyme. What is the role of that sigma factor in the transcription? With this question we will meet in my next session. Thank you very much.